Welcome to this Fast Facts presentation from Cell and Gene Therapy Insights and Centre for Breakthrough Medicines. I'm David McCall, Commissioning Editor of Cell and Gene Therapy Insights, and today we're discussing how to alleviate crucial challenges in scaling up high-yield AAV vector platforms. Joining me to give the presentation is Emily Moran from Centre for Breakthrough Medicines. Without further ado, I'll hand over to Emily to introduce herself.
Hi, my name is Emily Moran, and I'm the VP of Manufacturing here at the Center for Breakthrough Medicine. I have over 17 years of experience in clinical and commercial manufacturing, including cell and gene therapy programs such as AAV, Adeno, Lenti, HSV, and baculovirus systems. I've managed over 20 viral vector programs resulting in over 80 different batches of drug substance and drug product fills with a very high success rate. I'm here to talk to you today about alleviating crucial challenges in scaling up gene therapy manufacturing. We'll start with a little bit about CBM. So our mission is to save lives by accelerating the development and manufacturing of advanced therapies. At the Center for Breakthrough Medicine, we provide end-to-end platform solutions and services from concept to clinical and right into commercial manufacturing. Our capabilities include full-scale process development, cell therapy manufacturing, viral vector manufacturing, plasmid manufacturing, a full-scale analytical testing for our clients and for any external clients, and of course, R&D and regulatory services. Our site here in King of Prussia, Pennsylvania has 300,000 square feet manufacturing facility. Our team collectively has filed over 600 INDs throughout their careers. For Vector, our GMP suites, which are coming online this year, will have 20 plus available by 2025. And our testing facility is over 2,000 square feet of state-of-the-art technology to meet all of our testing and analytical needs. So by 2025, the FDA expects to receive upwards of 200 investigational new drug applications for cell and gene therapies. And that's annually continuing on. With an estimated approval of 10 to 20 therapies each year, this unprecedented growth comes with unique challenges for advanced therapy developers and manufacturers, which often center around three core must-haves: quality, speed, and flexibility. So prior to designing any purpose-built facility or selecting a purpose-built facility, it really starts with asking core questions about the vision that you have for your company or your product. What are your goals? What is your cash runway for what you need? And what are the things that you really want to define? So what are you aiming to achieve? There's many different advanced therapies out there in the market that require very different complex facilities. Deciding which ones are in the scope of the program or which ones are outside will really help you decide how you're going to build it. What is the strategic focus that we want to kind of focus on now and in the future? So if you consider some of the cash that you're looking at, do we want to save some of it now or build out all of the facility to support any level of product that you might see in the upcoming years? These are important questions to ask. And what other advanced therapies may you support in the future? What are the capacities? This can be very, very different, especially on the cell therapy side, which can be allogeneic or autologous, and that will really determine how the facility is set up. And how do you tie all these complexities together and remain innovative? So right now, we have certain things that we can do, but in the future, that might really change, especially with some of the new automated and digital advances in the field. And creating a timeless facility means being thoughtful about how we incorporate those innovative changes moving forward. And what challenges have you experienced in the past that you really want to avoid, or how do we make the operational support areas better and scalable for your business? How do we remain open to any modalities or technology that might be coming? But putting a purpose-built facility and answering these questions in front of any design is really going to streamline the activity. And designs have to balance a lot of module clean room efficiencies with configurability for all of your unique process needs. Our field is changing rapidly, which means the demand for processes and advanced therapies is going to change along with it, and they're moving at light speed. So having a facility that's very flexible to pivot to where that needs to be is something a lot of people must consider. There are multiple manufacturing scenarios that are timeless, that are sustainable, and some of them we've really kind of scaled out of that we don't want to build into our facility and limit the growth and the capability of that space moving forward. But it really starts with an intense level of preparation, as I spoke about earlier, and having that distinct set of questions so you really understand what do you want to be as a business now and in the future, and what do we need to be fast and flexible and always safe and really delivering the quality we need to meet our regulatory requirements, which means a seamless transition from clinical to commercial. So designing a facility simply for clinical might mean that you've missed very critical steps to sustain a regulatory inspection or make sure that that material is safe for the large commercial demand that it might face. Some of the ways that we do that is really with diverse equipment capabilities, and that creates a sustainable and agile future. A lot of the supply chain challenges we saw were a result of COVID and a result of a rapid growing advanced therapy space. And even though some of those things have slowed down, the future of our supply chain remains dynamic. So having diversity in the equipment that you select de-risks your supply chain, but it provides a lot of optionality in the future if something is hard to source after it's developed. It also creates great process characterization opportunities. So committing to, say, one type of bioreactor and not having the data for how your cells might perform in a different kind of bioreactor or what those settings might be could slow down future growth or future opportunities to pivot. So it really does create a great opportunity for you to get that data, see how the product or the process or the cells or whatever you're looking at react in that set of technology and how it might react in another one. If you have to pivot to an outsourcing model later on, you'll have that data at your fingertips and know exactly what you need to do to transition into a different technology or a different area. And it really de-risks the supply chain, not only for your product, but for the future patients that need to get that material at clinic. Considering having a pilot process development lab or full-scale process development capabilities provides a safe space to do all those comparabilities, studies, and provides valuable insight into the future. Early investment in process definition really de-risks it because you won't get siloed or put into a corner with the type of technology or the type of process that you need to do. Establishing robust control limits and key process indicators on different technology platforms will just help you further understand what are the areas that you can pivot to have a more robust and optimized process, increasing yield and reducing cost of goods year over year as you develop each step. For example, here at the Center for Breakthrough Medicines, we got a variety of large-scale bioreactors from multiple different vendors so that we could do characterization studies of cell lines in different units. We have that data as a library to offer to our clients in the future so they can understand how similar cell lines will produce in that technology. We also have multiple different downstream options for both TFF, chromatography, and of course, ultracentrifugation so that we understand the best way to get that empty versus full separation, as well as optimal yield and recovery when reducing any kind of burden we don't want to see from the product. Great development comes from great development spaces. So how do you pave that way for high yielding and cost-effective vector production? A lot of it starts in how we designed our process development space. The first thing that I'd like to point out is our process development space is on the right side of our screen, but on the left side, it's connected to our analytical development and testing area. This allows us to identify results, in-process testing, and any other kind of analytics that we would need to hone in on the best process possible. Each one of our rooms is designed for a different scale of the process, going from very small scale, less than a liter production with our high-speed Amber 250, all the way through our full-scale bioreactors, right now going up to 500 liters with an ambition of 1,000 liters in the near future. It gives us that safe space to really practice how we would scale up and design all of our programs and all of our experiments to bring optimal yield and cost-effective production for each program. It's over 5,000 square feet of production harvest purification, as well as formulation, fill, and full drug product capabilities here on our campus. We keep these things sort of segregated so we can decouple unit operation development from one another and safeguard all of our IP as we have multiple different programs in process. All of our technology is cutting edge, and it benefits each person in this process to adopt new technology or at least test it or provide a space that we can test it so you're aware if it's a fit for your process. I'd like to go through a case study in which our pilot operations lab in our PD space was the main reason for our success. CBM formulated a flexible, client-specific way of working and service to meet their client objectives. And that's really one of our core cultural components at CBM is having a true partnership and being that process development and manufacturing arm of all of our programs. The need here was preclinical AAV generation, purified drug substance at a 200-liter scale. There was a rapid desire to get it into production in the pilot space to meet IND-enabling activities. The challenges that we faced as a team were procuring equipment and materials to meet this incredibly tight timeline in our strapped supply chain environment. Ongoing, our process development meant we needed to evolve during the tech transfer process and kind of iteratively say, what is the most robust process and how do we make sure that we're meeting our deadline to start pilot operations? We aligned on our goals. We had extensive ongoing collaboration between the SMEs here at CBM and our client program. We identified equipment that represented our biggest risk, what we had in-house, what we needed to get rapidly, and what material gaps existed that we could close or that we needed rework around. What bespoke manifolds or filter assemblies or transfer lines were we going to have to create to make sure that we hit our timeline? We did that together. We trained at CBM and visited our client site to make sure that we were lockstep in our execution and kicked it off within 1 week after signature. Our tech transfer was completed end-to-end in 2.5 months with all of our risks managed. We executed the pilot within the typical time frame, and this led to a right first time execution of a 200-liter AAV in less than 4 months start to finish. Regulatory guidelines are really the North Star to achieve success. So as we design any program or facility for a scalable AAV process or beyond, it's important to understand what the regulatory and safety requirements are for that process and that facility from the get-go. Upfront investment into engineering controls really allows for appropriate segregation and will pay dividends in a multi-product facility. So although we talk about scaling AAV and getting that to market as the focus of this presentation, many CDMOs are challenged with other molecules beyond an AAV, and it's important to design a facility that can safely segregate that production in both process development, testing, and of course, GMP. We mapped out all of the procedural controls that we needed, from putting on a latex glove to moving in a bioreactor to ensure it was optimized for the personnel, the equipment, and of course the waste flow, which often represents the highest risk in the facility. Taking a strictly clinical approach might require costly rework later, as HVAC considerations are quite expensive. So for our facility, we designed 100% single pass air as our HVAC strategy to prevent contamination, unidirectional flow with segregated airlocks for both material, personnel, and waste, and a very robust contamination control strategy, which involved normal disinfectants, engineering controls, as well as VHP decontamination after every batch. A case study here where we have 2 scenarios and how we designed it is our downstream processing areas. We didn't want to compromise with what might be required, so we designed one of our suites to accommodate grade C or closed system processing and another suite to really accommodate more open centrifugation-based processing. We designed this space with more engineering controls to make sure that we presented a safe space for the operators to work in when extracting some of the material, as well as any space that might not have sterile filtration as an option in the program. By designing this flexible space for each one of our potential programs, we never had to say no to a client, and we also did not have to do any costly rework to accommodate a program and allowed us to get into our GMP area much faster. And never forget our optimizing enabling functions. We talked a lot today about process considerations for scale up, but any good process needs a good support system around it. Having buffer and media preparation areas for those custom materials, having a material and decontamination transfer strategy in the facility is key, and being able to build any customized components, filter assemblies, or anything else that might be specific to the process and hard to source is very, very important. And of course, every drug substance must go through a drug product before it gets to that next step. Having drug product on site is something that everyone should consider in both their outsourcing and in-house strategies. Having good drug product capabilities increases your speed to clinic and make sure that you have maximum doses for each patient. Quickly, I wanted to give an overview. We were very fortunate to be able to establish CBM here in beautiful King of Prussia, Pennsylvania at our 411 site. This site offers multiple buildings so that we're able to segregate all of our unit operation and testing area, as well as gives us ample space in the back half there in yellow to expand both internally or externally for any one of our partners right here on our site. This included a thoughtful approach to waste permitting and other capital investment structures to make sure that we could safely expand it here on space. I want to thank you for your time today and listening to our presentation. I wish everyone the best of luck in their scale up and look forward to answering any questions that we might have moving forward. Have a great day.
Well, thank you very much indeed, Emily, for a fascinating presentation. And thanks to you, our audience, for watching today. If you'd like to download a poster version of today's talk summarising the main points, you can find that below the video. We hope you'll join us again soon for more Fast Facts.